So welcome to Synthace's DOE Masterclass Part 2. My name is Khaled Elawad. I'm a Scientific Solutions Consultant here at Synthace, and I'll be your host for today's session. If you've attended our first installment of the DOE Masterclass, welcome back. And if you're joining us for the first time, we're very excited to have you. Can't wait to share today's session with you all. Before we get started with the presentation, I just want to explain the format that are going to help you get the most out of this session. Please make sure that your Zoom settings are set to computer audio only. And should you have any technical difficulties, send us a private message in the chat box at the bottom of your screen. This webinar is being recorded and will be emailed to you afterwards, as well as made available on our website to watch on demand. We encourage that you take advantage of this interactive session and ask questions into the Q&A box at the bottom right of your screen. We're going to answer these questions during the second half of the webinar, but please feel free to add them in at any time during the presentation. So as I mentioned before, this is the second installment of our Masterclass series. There's a huge amount of depth and detail to DOE, and we won't be able to cover it all in one session. So our first session started with an introduction to DOE and showed you the power that it can have to transform your experimentation. You can catch that on demand on our website. Today's session will walk through detailed real-life examples of where we've used DOE, and the third session coming next month will show you how to perform DOE in a pragmatic way. By the end of this three-part intro series, you'll have all the starting tools you need to run your own DOE. So if you haven't done so already, please sign up for our third webinar. The link to do so is going to be in the chat. And keep an eye out for any future webinars that we have in this series. So now I'm excited to introduce our presenter for today's webinar, Dr. James Jaja Arpino. As one of the foremost experts on DOE at Synthace, he's an evangelist for transformational multifactorial methods in biology, and Jaja has over 10 years of experience in DOE, and his background blends both theoretical and practical applications of DOE to support life sciences research and development. So without further ado, I'll hand it over to today's speaker, Jaja.
Thanks very much, Khalid, and thank you to everyone who's joining us again for the second part of our DOE Masterclass for Biologists. I'll start with a quick apology that I hope the glare of the lights off the top of my head doesn't blind anyone. And with that, let's jump into some DOE. What I want to do, first of all, is just start with a quick recap of Marcus's DOE Masterclass, the first part. And we'll recover some of the very high level differences between one factor at a time and DOE to get everyone warmed back up for this session. So as Marcus presented previously, if you attended, and if you don't, then hopefully you'll gather what I'm trying to get across here, the differences between one factor at a time and DOE. Biology is an emergent field, and as such, like the classical one factor at a time approaches to studying the biological systems that we're all working with in modern day, they're really not suited for the high level of complexity that these biological systems have. A very quick, simple example of this is, imagine we're looking at two different factors that we may want to investigate in a biological system. In this example, temperature or pH. And imagine, first of all, we fix pH at a specific setting, and we vary temperature from 15 degrees to 35 degrees here in this example. And we collect some data and we find out where the optimum of that particular factor lies. So for temperature here, it's somewhere right in the middle of 15 and 35 degrees. Then we decide to fix that factor at that level, and we vary our pH factor. And we collect our data from that particular experiment, and we start to find an area of our landscape or experimental landscape where we think that's where the optimum sits. But of course, what we miss with this sort of experimentation is that we don't see any of the interactions that might be taking place between those two factors, temperature and pH. And so whilst we think we've found an optima, actually our optima may sit somewhere else further out of our experimental landscape, and we've missed that interaction. So we haven't learned anything about how these two factors impact one another. But this is obviously a very simple system. It's only two factors that we're looking at here. Imagine actually we wanted to investigate, let's say, 6 factors. And here we've crudely represented a 6-dimensional space in a 2D diagram. So now let's say we've got factors of temperature, pH, and time, but actually we've got 3 more factors, A, B, and C in the system that we want to investigate as well. If we wanted to learn absolutely everything about this particular biological system and how these 6 different factors impact that biological system, then we would need to do every single one of those experiments, or what we would call a full factorial experiment. So the question sort of is asked, well, why not? Why don't we just do these brute force approaches of full factorials? Well, hopefully the numbers here will start to speak to you as to why that isn't really a smart way for us to move forwards or to do designs in that way. So for a simple two level, 3-factor design, so think about this large cube in the middle, you're looking at doing 8 experiments. So an experiment at each one of the vertices. And then the example that I'm showing here, we've got 6 factors with 2 levels, a low and a high set point for each one of those. That would take 64 experiments. But really, we're trying to push the boundaries of what we want to study with these complex biological systems. And we're looking upwards of 10 factors to 15 factors. Well, you start to see that the numbers of experiments that you would need to do in a full factorial start to explode exponentially. And that's only if we're looking at 2 levels at a time for any one of our factors. Let's say we actually want to look at a low, medium, and high set point for any one of our factors. So we've got 3 levels for, let's say, 15 different factors we want to investigate. Well, you're sort of now starting to look in the millions of experiments to be able to capture that entire space. So where does DOE actually come in? Well, DOE is the statistical framework that allows us to pare down all of those different combinations of factors and levels that you might have in a full factorial down to the few experiments that you would need to do to answer the main questions that you're asking about your biological system. That might be, which of my factors are having main effects, so that they're impacting the system alone, maybe 2-factor interactions, 3-factor interactions, or more complex. And the way we do that is with various different DOE designs. And there are lots of different DOE designs out there. And I think often this is where a lot of biologists, even at the very get-go out the gate, we can get unstuck. We start to feel overwhelmed by the number of different designs that are on offer to us and which ones we should be picking when, what are they useful for. And so I'm going to try and touch on some of that today at a high level. I won't dig into the details of each one of these designs, but we'll go through 4 of these designs today, examples of them. And hopefully we'll give you a feeling for when you might pick some designs over others. For those of you who attended Marcus's first talk and you weren't convinced about what Marcus was saying about DOE and versus a 1-factor at a time approach, or for those who have just heard what I've had to say and you're still thinking, oh, I haven't got enough information here to decide whether DOE is useful to me or is something that I actually want to start doing, I would really love to implore all of you to go and read this absolutely excellent review paper. They really break down the fundamentals of why DOE is a much smarter route to go down than 1-factor at a time. And so please, if you have the time, go and read this paper. It's absolutely fantastic. I will dig into very briefly one of the figures that they present in this paper to hopefully, again, speak to this, why we want to do DOE versus 1-factor at a time approach. So imagine you've got 2 different laboratories, Lab A and Lab B, and they're both investigating the same biological system. And I would be very surprised if any of our attendees today haven't read multiple research papers out in the field of the same biological system, and the 2 research papers give conflicting results. And you might be thinking, well, which one is the correct one? Well, both of them may be equally correct. It's just the way that the experiment was done that you're lacking the information to fully understand the system. So in Lab A, they investigate factor 1, and they see that increasing factor 1 decreases the response that they're interested in. Whereas Lab B, they vary factor 1 and they see an increasing response to the system. They have all the data that proves that they are both correct. They're both equally valid. So let's have a look at if you treated this as a 1-factor at a time approach, and you were actually aware that there is this second factor in your system that gives rise to the differences that the 2 labs were looking at. But imagine now, as a 1-factor at a time approach, Lab A decides to investigate factor 1 first, and they see that it decreases the response with increasing the factor 1 levels. They then fix their factor 1 level at that first concentration or factor 1 at the first level, and they then vary factor 2, and they see that the increasing factor 2 or decreasing factor 2 decreases the response. Likewise, Lab B do the opposite. They test factor 1 and they increase the response, and they then fix at that high factor 1 set point, then look at factor 2, and they see that that decreases the response. Again, both of these labs have validated their own sort of self-fulfilling prophecy if you like, that they are both correct. They've both learned about the system, about factor 1 and factor 2, but they've come to very different condition sets that describe that system. And I think this speaks a lot to some of the reproducibility crisis that we have in the biological R&D domain space when it comes to reading literature and trying to understand how do we reproduce experiments when we've got these very different condition sets being reported. If we have a look at what DOE might be able to offer you, in just 5 experiments, either of those labs would have been able to get a full understanding of the entire experimental landscape here and being able to derive the contour map that you see against this particular figure. And so you can see that with fewer experiments, you can get much deeper insight and better understanding and learnings about how your biological system behaves. So as Marcus spoke to previously, DOE is an iterative process, but how you decide to progress from iteration to iteration, it's not a cookie cutter process. There's no one fixed method that you follow, and it really depends on the type of experiments you're doing, what you're trying to learn, what you're trying to achieve, how much knowledge you have up front, as to how you might go through your iterative DOE strategy. Today, I'm going to try and walk us through 3 very different strategies that and speak to when and why you might go one route or another. And so as Marcus touched, you have sort of factor screening stages where you're looking to determine which factors are most important. So you start off with lots of factors, which ones are having the most impact on my system. The ones that aren't, we can sort of put to the side and take the ones which are having an impact forwards. Iterating is when we're moving to a better design space. So we're looking at, out of those factors we're deciding to keep, which ones we're having the desired effect on our response. And we'll start to move the ranges of those factors to hone in on the area where we have the best conditions. Then we might have refinement stages where we start to look at slightly denser designs. We start to look at for curvature in the response landscape to our different factors. We get a deeper understanding of the factor interactions that are taking place. And then you may have an optimization or robustness stage at the end of your strategy, which sort of allows you to hone in on the most optimum conditions. You can often, with high quality data, generate very good predictive models at this stage. And you can really understand how robust those conditions are to perturbations in the system. So if you were going to pick one of your conditions to take forwards, how robust is that to sort of changes in room temperature, for instance? So there are different stages of DOE and they can be applied in different ways. But as I said, it's not cookie cutter and there's no one approach to doing this. You have the freedom to apply these different stages as you see fit, depending on the system that you're working on. So again, a quick reminder on what is the DOE process for any one of those iterations. Well, we sort of boil it down to 5 main steps. You often start with gathering as much knowledge about the system that you want to study. So that could be literature mining, speaking with experts, your colleagues, but the idea is getting as much information about the system that you want to study, and also deciding what it is that you want to learn from the system. So what is it you're trying to improve? Are you trying to maximize yield? Are you trying to minimize noise? So setting your objectives for what you're trying to do. The next step is just choosing a design. So, I mean, we do this every time we plan an experiment. We design how we're going to set up the experiment, which liquids we're going to use, how we're going to pipette them. So you start designing the experiment you're going to do. Third step, you're just going to get in the lab, run your experiments. It's everyone's bread and butter, right? We all know how to do that step. The next step is gathering your data. So you're going to go to your analytical device. You're going to measure your experiments. You're going to generate some data. You're going to structure that data. You're then going to do some data manipulation on it. So raw data often is meaningless to us, but we want to maybe do a baseline correction. Then we want to do a normalization against a baseline, or we may want to do an interpolation from a standard curve to get a concentration value. What we're interested in at this stage is the biologically meaningful value that we can derive from that raw data. Finally, we may build some models, use statistics, derive insight from the data that we've collected. So, as I said, none of this is different from a standard experimental process, taking DOE out of the picture. So where does DOE actually come in throughout this process? Well, it comes in at step 2 and consequently step 5. So it's choosing the design. It's deciding which experiments am I going to set up to give me as much information as possible to answer the questions that I want to ask about my system. And consequently, the models that you might fit to that data downstream are quite heavily dependent on the design that you chose up front. So it can be quite a logical process to go through for DOE. Marcus took us all through a case study of the expression of a human enzyme carboxylesterase in Escherichia in E. coli. And we looked at genetic factors such as sort of ribosome binding strengths and plasmid copy numbers, looked at process factors, temperature, time, media factors, so inducer concentrations and base media that we used. And as you can see, for each of the different colors, sections of the bar chart, we went through an iterative process, starting with a screening design, going through some refinement designs, and finally an optimization stage before validating to identify some really robust conditions that were giving us a 200-fold improvement on our yields. These are the type of things that you can expect to get from DOE. And certainly when I joined Synthace and was first introduced to DOE, I was skeptical about it until I started seeing data like this. And that's what we want to do in today's session, is to take you through more case studies of slightly different flavors, and we'll speak to the different designs that we used and the different strategies that we used. And hopefully you'll start to understand that DOE is a really flexible framework that you can apply to your research. Before we jump into that new content, you might recognize this this poll from the first webinar if you attended then, and we just want to do another pulse check for where everyone is on our DOE journey. So how convinced are you that you need to use multifactorial methods in your biological research? The poll should be open for you now, so please go ahead and vote. And we'll give you a couple of minutes to do that. And just a reminder from last time when we asked this poll, about 36% of our attendees said they had heard of DOE. Sounds potentially useful. 34% of our attendees were completely convinced by DOE, and they were here to learn about more material and convince others. 17% of us were quite bought in, but still had some reservations. 13% didn't have an opinion. They were still here to learn. Amazingly, nobody that attended were fully skeptical. So let's see if we've made any changes to those this time around. So at the moment, it's looking like 50% of the people are convinced and about 21% are quite bought in. So it looks like the numbers are heading in the right direction. We're starting to convince you of DOE being the methodology that you should be using or want to use in your biological research. And I think we're going to wrap up the poll now. And yep, as I said, 50% people are looking convinced, 21% of us are quite bought in, 25% have heard of DOE, and 5% of us still don't have an opinion. So hopefully at the end of this presentation, we can get the 5 of us who don't have an opinion up into those top bracket categories. And amazingly, we still don't have any skeptics, which is absolutely fantastic. So moving on, let's jump into a few real world applications of DOE. As I sort of mentioned previously, you know, the underlying statistics of DOE can be quite heavy at times. And I think certainly when I first was introduced to DOE, as I said, that I was a skeptic before I started seeing the results of what could really be achieved with it. I did find the barrier of entry to learning DOE quite steep. And I think it's because a lot of the training materials out there, they really throw you in at the deep end with the heavy statistics quite early on. And it can often be quite difficult to sort of get traction and know where to start. We wanted to try and take a slightly different approach with this webinar series, and we want to take a slightly gentler route into the world of DOE. And we want to sort of focus on the real world use cases first, showing some data, how we applied DOE, and touching on some simpler guidelines. So not rules, but guidelines that can help you start to formulate your own ideas about what you might want to use DOE for and how you might do that. Some tips and tricks about looking at multifactorial data, and to get a better understanding about where it can be applied before we go deeper into any of the technical details. So the applications that we want to go through today, the applications that we've got are sort of, and examples that we've got, the case studies are focused around sort of the bioprocess value chain. So we have, strain or cell line engineering / development, upstream cell culture, downstream purification, formulation development, and maybe assay development. The examples we're going to go through today are construct assembly, so a genetic construct assembly example, which you might use for upstream strain engineering or strain development, a transfection and transduction optimization, which sort of speaks to our cell line development process where you need to get DNA into your mammalian cell lines. And we're also going to focus on an assay optimization, or more to say an assay characterization. But we have examples across the board in these different columns that you're seeing now. Formulation development, as you can see, is pointing to media development, very, very similar use cases where you're playing around with the components of a formulation or a media. One thing I want to point out is the transformation development in the cell line development column, something that I'm not going to go into today, but I'll speak to very briefly coming up. Competent cell development, we've got examples of as well. So for all you people out there working with some weird and wonderful new organisms, but you maybe don't have the molecular biology toolkits that are available to a lot of us who are working in E. coli, yeast, or mammalian cell systems, so more standard systems. But DOE could be a real game changer for you to better understand the conditions required for efficient gene transfer into those new organisms, to start building up that toolbox to be able to work with these new novel organisms. Similarly, with DNA library generation, we've got examples there that speak to, what might you want to vary to be able to improve coverage of your libraries whilst minimizing bias in those libraries as well. For downstream purification, we've got, again, great examples around optimizing antibody purification in miniaturized purification columns on automation platforms. So if anyone here is interested in any of those ones in red that we're not going to be covering today, please feel free to reach out to us and we'll be more than happy to go into details around those for you. So I'm going to start up front with different types of DOE designs that you'll be seeing that have been used in the case studies that we'll go into a little bit later. So some of these you may have seen in the list earlier on I showed, but we've got two-level fractional factorials with some midpoints in there, central composite designs, and space filling designs. So we've got three more common DOE designs that you see being used, and one less conventional, the space filling DOE design, you don't often see in the biological space, but I'll speak to that a little bit more later. Again, I won't be going into heavy detail on how these designs actually work underneath the hood, but if there is interest, please do reach out to us again, and we're more than happy to speak to you about that. So two-level fractional factorial designs, they're the sort of designs that you see at the factor screening level. So this is where you're really starting your journey. You have all these ideas of all the factors that might be impacting your system, and you want to very quickly sort of filter out the ones that aren't and take forwards the ones which are. And so you might use a design like this for screening those factors and starting to get an idea if there are factor interactions in your system as well. The two-level fractional factorial with midpoints, it's the same design, but with what we call sort of center points. So you'll have a set of runs in your design which take on the middle level of any one of your factors. So you'll have a set of runs that sit in the very center of your design. Again, these are really great for screening factors still, but sort of refining your space, getting an indication of whether there's any curvature in your response landscape to for any of your factors. And also getting a deeper understanding again of the factor interactions. Central composite designs are a type of design that are response surfaces. And really we use these for sort of doing optimization or robustness studies. They are much more detailed, give you a much more detailed understanding of the curvature in your design landscape. And they give you really detailed understanding of the factor interactions that are taking place. And these are the sorts of designs that you might use a little bit later on in your DOE strategy. And they often can yield high quality models that can be used for prediction of the behavior of your system as well. Space filling designs, the more unconventional design type that you don't often see in biology, they can be brilliant for screening and optimization, depending on how many runs and your capacity that you have. They're brilliant for systems where you have lack or no prior knowledge of the system that you're looking to optimize. So if you don't have a starting point, you don't know, what your levels of your factors should be. They're also really good at being able to identify nonlinear landscapes in your experimental space as well. So again, I want to introduce the idea of how do you know what to do next when you're going from one iteration, so you've done one design in one iteration, to the next iteration. So how do you actually derive inference from your data and decide, well, what should I do next? What's the next design? Where should I move my factor levels to? And this is a very, very simple example. I want to introduce it now because it might help you to understand as we go through the case studies how some of the decisions were made about going from iteration to iteration. As I said, very simple, one-dimensional use case. In reality, DOE is a little bit more complex as we work in a multi-dimensional space, but the premise of how you might interpret the results is still the same. And the data shouldn't scare anybody when we think about multi-dimensional data sets. We're very good at working with up to 3 dimensions, maybe 4 dimensions. When you're getting up to 10 or 12 dimensions, I think it can sometimes start to feel overwhelming, and you can feel paralyzed not knowing what to do next, or even how to start looking at that data. What I'm going to show you here are just guidelines. They're not rules, but they are things I always find really useful before I ever fit any models to my data, and I am just eyeballing my data for the first time. Never underestimate the power of eyeballing your data before you start fitting models and driving inference that way. So imagine we've observed a factor interaction. There are 3 different types, an effect that a factor is having on our response. And there might be 3 different effects that we might see. We might see a positive effect. So as the factor concentration increases, our response increases. We may see no change at all. And then we may see a decrease in our response as the factor increases. So before I speak about the first or the third situation here, the middle one, just because you see a flat line here, it doesn't mean that the factor is not having an impact. There are chances that it could be, but you started with your high and low set points in the wrong space and you're missing the area of activity. Remember, in these sort of starting fractional factorial designs, we often only have 2 levels to a factor, a low and a high. And so you might miss that activity window and think nothing's happening, but it could be. And this is where I want to sort of remind everyone that we are still scientists, and you still need to use your scientific gut feeling and knowledge that you have about the system, that if ever you see something that is flat like that, but you really were expecting it to have an impact, maybe you don't want to discard that factor, but keep it in your factor set for the next iteration where you might be able to get more information about it. For the next set of graphs, I'm going to focus on this first example here. So where we've got an increasing response with increasing factor concentration. Now, with 2 data points, we can only ever fit a straight line through these to understand what's maybe happening in our system. And as we fit models to these 2 level designs, I want to remind everything of something that George Box said, and I'm sure everyone will have heard this, that all models are wrong, but some are useful. And again, this sort of speaks back to using your scientific intuition and being a scientist and not putting all your faith into models and statistics. They can be equally misleading as they can be useful. So there's a certain amount that you can learn from models, but before you get going with modeling, let's just look at data. So if we've got 2 data points and we fit a potential straight line through those, let's hypothesize what might be actually happening in our system if we had more data points. Now, we may have it right, and it is just a linear interaction here, a linear effect. And our next data point might be fit in that line perfectly. But equally, that next data point could sit in very different places, and our data could be a very, very different shape to what we think it might be, given that straight line that we fit through those 2 data points. So what do we do in the next iteration? What might we do? Well, imagine if we took our low and our high set point and we just shifted them further up on our scale. So we move everything up to a higher concentration. No matter what the shape of our data actually is under the hood, just by making that shift of the low and the high set point higher up, we're able to capture any one of those behaviors if they're happening. We will be able to sort of interpolate and understand exactly, like from the first iteration to the second iteration, what our data might be looking like under the hood. Similarly, with that negative effect of a factor increasing and the response decreasing, you might, in the same instance, shift your low and high set points lower in the range, for instance. And again, it depends on what your objective of your experiment is. Is your objective to maximize your response, or are you looking to minimize the response? And that depends on the response that you're measuring. So you might want to minimize noise, you might want to maximize yield. But shifting your low and high set points, you can really sort of get a feel from just looking at your data, what you should be maybe doing next. And on that note, let's jump into our first case study. So the construct assembly optimization. Genetic construct assembly, this is a Golden Gate assembly method. And the objective for this particular case study for us was to get a robust method that we could run on automation platforms, was transferable across different automation platforms. So we weren't optimizing the different genetic parts that we were putting into the constructs, although you of course could do that if you were looking to sort of do optimizations or characterization of genetic networks, for instance. But for us, we were looking at the process. So we were very focused on process parameters here. And an objective for us was for the end process to be as simple as possible to run as well. And so you can see that we went through a strategy of initial screening. We did a refinement DOE, we did a second refinement, we did a validation, and then we tested that the final condition set across multiple different automation platforms. Some tables here showing how we navigated through the design space, and I'll go into those in more detail. So firstly, how do we visualize multi-dimensional data? Imagine you haven't fit a model to this data that isn't guiding you to the effects that you are interested in, or any factor interactions that you might be seeing. So let's just have a look at this data as if we were eyeballing it. So we can plot a graph here looking at two dimensions. So our incubation temperature and the concentration of DNA in the reaction. And I can see that high concentration, high temperature, I'm starting to see a certain number of colonies that we're getting from this particular experiment. But then let's look at the third dimension and the fourth dimension as we bring these in. And you can see that with incubation time, okay, now I'm starting to see something really interesting here. So long incubation times at higher temperatures with higher DNA concentration, I'm starting to get a lot more colonies. But then if I was to do a cycling process in a thermocycler for this particular experiment, I can see I actually start to see this tighten up, the data tighten up. It's getting a bit more robust. But of course, this isn't the only response I might want to look at. I might have more than one response that I'm measuring. And in this particular example, we were measuring the number of colonies that we're getting back after transformation, but we were also looking at the percentage of those colonies that had the correct genetic construct in at the end. And if we look at those same four dimensions, but for our second response, we can start to see that there is some really compelling evidence that we've got some conditions here that are giving us a very high efficiency. And so just looking at this data, we can start to sort of identify things that we want to fix for our next DOE. We're not going to change in our next DOE design, and some areas where we want to change the ranges for these particular experiments. As I said, this initial DOE was a screen, and we used a two-level fractional factorial. So a design where we only have a low and a high set point for each one of our factors. We're fitting linear models to this data and understanding what impact the factors are having. So from this particular experiment, we were able to say that, yes, we think cycling is having a real impact here, especially when we're looking at the efficiency. It's really tightening things up. So we decided to, yes, we're going to keep cycling in our next iteration, but let's dive into the details of that cycling. Let's actually introduce two new factors of how long it takes, how long we leave the experiment to cut the DNA and how long we leave it to stick back together, so the ligation. We fixed the temperature, the concentration, the buffer as well. So whilst I haven't shown buffer in this example, we actually identified from the data the CutSmart buffer with additional ATP was slightly better than the ligase. But you've always got to remember, well, what was the objective? And our objective was to have a simple system. And whilst the T4 ligase maybe wasn't, T4 ligase buffer maybe wasn't quite as good, it was good enough that we felt like it simplified the process if we weren't having to handle more than one component during the setup of the experiments. Some of these other ranges we started shifting to investigate slightly different areas of our space. So in the next iteration, those experiments that we did, focusing in on the cycling condition, so the cutting time and the ligation time, we saw that by increasing the cutting time and the ligation time, you really start to see a dramatic decrease in the number of colonies that we were getting. but still in the order of 250. And at the end of the day, with genetic construct assembly, you only need one colony to have the correct construct in. So we're still in a regime where we haven't hit failure. We're still getting plenty of colonies, so we're happy. But the interesting part of this data really was actually the number of correct colonies we got from the different cycling regimes that we tested. And what we could really see was that longer cutting times and longer ligation times gave us much more robust assembly conditions. So again, just by eyeballing this data, we can start to say, okay, do you know what? I'm going to fix my my cycling. We're going to do it, and we're going to fix the time at 5 minutes and 5 minutes for our cutting and ligation, which was giving us the most robust conditions. We tried denaturation of these reactions before transforming them. Literature had identified that maybe that could improve the efficiency of the reactions. We didn't feel like that it really gave us much difference at all across the experiment at the end of the day. So we actually dropped that as a factor. Again, started fixing things like the ligase concentration. We weren't seeing an effect from that, so let's fix that at the lower level. So move on to the final iteration that we did. And now you're really boiled down to only a few factors here. We've only got 4 factors and, you know, simple plots here. We're only looking at 3 different dimensions. And what we can actually see here is that the concentration of the enzyme, of our cutting enzyme, was having no impact on the data at all. But the concentration of the DNA parts in the reactions versus the number of DNA parts we had in the reaction was having a big impact. So if you've got lots of DNA parts, go with higher concentrations of your DNA. With far fewer DNA parts, you could get away with lower concentrations of DNA. Again, we fixed those conditions at the high concentrations, and we now had a set of factors that were fixed across the board. And what we wanted to do was validate this particular set of conditions against a whole host of different numbers of DNA parts. And when we performed that validation, we did it against different genetic backgrounds as well. So we did 4 and 5 parts assembly with a COMET GFP genetic background, 6 parts assembly with a ZAL-T reporter construct, 7 parts assembly with an EGFP genetic construct, and 8 and 9 parts, again, with a ZAL-T reporter system. And you can see we had really robust final working conditions that were always giving us colonies, were always giving us the correct colonies when we screened. And that particular condition set was then transferable across multiple different liquid handling automation platforms for us. So we ended up with a really robust system after 3 iterations in the validation runs. So that was the first case study. Let's jump into the second case study. So this was transfection and transduction optimization, and this work was all carried out by our partners, Oxford Biomedica, and really cool system. They were looking to optimize their transduction efficiency and robustness. But the way they were trying to do that was actually by optimizing the transfection conditions that give rise to the production of the viral vector that they then use to do the transduction step downstream. And so the sorts of factors that they were looking at were the various different DNA components going into the reaction, the transfection reagents, chemical additives in the system. And what I want to briefly touch on is the actual strategy, the DOE strategy. So this sort of standard strategy we spoke to before, the typical one where there's a factor screen, an iteration, a refinement, and an optimization. Oxford Biomedica felt that with the power of automation, allowing them to do many more runs in a particular design, they could actually skip some of these steps and go from an initial factor screen straight into a refinement and optimization design without needing to do the intermediate steps. They could get a lot more knowledge from larger, more complex designs. And so in their first iteration, they had a set of factors that they were investigating. And here's an example of where not just eyeballing the data, but actually fitting a model to the data. There are many tools out there and different statistical tests that can use to guide you towards where you should be looking in your data for where there are interactions. So a half normal plot like this, where any of these points that sit on the blue line is in the noise of your system, and everything that's in red are signals that you're seeing outside of the noise of the system. And this is a good indication that those things are having a real effect. And some cool ones to pick out here were 2 of the DNA factors, if they were put into the reaction in conjunction, completely nuked the transfection and transduction efficiency. Whereas having one or the other in the system, everything was working absolutely fantastic. So one of these was performing slightly better than the other. So DNA 5 was dropped out of the system, for instance. Again, 2-level fractional factorial design used for the factor screening. So again, you can start to see common themes here of fractional factorial designs used for screening factors. And again, 2 levels, when you fit a model, you can only fit straight lines through those 2 data points. So you start to get an estimation of your experimental landscape, but you don't have enough detail to truly know necessarily what's going on here. So in the next iteration, Oxford Biomedica chose to use a central composite design. So this is one of our response surface designs where you're looking at a much more detailed, denser design with axial points levels. So you're looking at 5 levels for each one of your factors. And that gives you a really high level of detail to be able to fit higher order models that give you curvature or show you curvature in your system. And that's sort of exemplified here with these model simulations. Showing where the curvature in the system is. Again, as I said, DNA 5 was chucked out and the cell concentration was fixed to this iteration, learnings from the previous iteration. Some of these ranges shifted up, some of them shifted down based on the data that we saw in the previous iteration. And ultimately what they were able to achieve was a 10-fold increase in the viral titer and up to an 81% reduction in the pure error. And this was really the thing that they were after, is that robust process that they could run over and over again and know they were always going to get great transduction efficiencies. Finally, let's jump into the last case study, and this is the transaminase activity characterization. So transaminases are enzymes that transfer amino groups between different compounds, and they're used heavily in as biocatalysts for the production of valuable products in the pharmaceutical industry. And this particular enzyme was converting 2 substrates, sodium pyruvate in MBA, into acetophenone and sodium alanine, the product that we're interested in this particular system. So what sort of factors might you want to look at in a system like this? Well, we've got the substrate concentrations, maybe enzyme concentration. There's a cofactor involved in this system, so let's have a look at the concentration of that. What about if we wanted to investigate whether there was product inhibition in this system? So maybe we should include product in our experiments as well to get a better understanding of the mechanism of our enzyme. Then some sort of buffer components here. So salt type, the salt concentration, the solvent concentration, if maybe you need solvent to solubilize your substrates. Then some process parameters, so temperature and pH, the buffer type that we use, and then looking at the enzyme stability over time in these reaction conditions as well. So this is where we started to think about using the non-conventional DOE design like space-filling DOEs. And why might you want to use a design like this? So in the assay space, where most experiments are being carried out on 384 or 1536 well plates, and you normally have access, or we might have access to automation to set up these experiments, the space-filling DOE becomes a really attractive type of design for looking at this sort of assay space in very high resolution, when you have very little prior knowledge of the system as well. So let's just have a look at the example of a traditional DOE, so a fractional factorial, let's say, and we pick our low and high set points. And as I was saying earlier, you might miss that activity window of your response because you set your low point too low and your high point too high, and we miss any activity here. So you might think that that factor is not having an impact on your system, whereas in reality it actually is. With a space-filling design, you have a very granular set of levels for any one factor. And so you get this really highly detailed presentation of your response landscape. So what does a space-filling design look like? And this might speak a little bit more to why these work really well in spaces where you have access to automation. So these are the factors and the levels that we wanted to investigate in our space-filling DOE design. And if we look at this particular plot on the right, I know all of the labels are too small to read, but any one of these axes is one of the factors and their levels crossed against any one of the factors and their levels. So we're looking at an 11-dimensional space here in 2D. Each one of the black dots is another experiment that you are setting up. And you can see that these are such complex designs. They're not the sort of design that you would likely set up manually in the lab. But with automation, they can be extremely powerful designs. You can learn a lot in a single iteration. So in this single iteration, again, we fit a model to the data. Again, we've got a statistical plot here showing the measured yield that we got from our experiments against the predicted yield from the model. So we can see we actually have a great correlation here that the model we fit is describing the experimental data very well. There are then tools available to you in third-party statistical softwares that allow you to sort of investigate that model in a sort of simulated way. So we call these prediction profilers. you can drag these red dots any direction on any of these graphs, and you'll see all of your other factors change, and the responses to those other factors change as you change your different factor levels. They can be hugely useful and insightful at directing you towards areas of activity, factor interactions, etc. And so from this, we can get an idea of whether there is any product inhibition, as I said, by including product. In this case, there wasn't. But there are some very clear standout effects that we can see from some of the factors in this particular landscape. And what we can generate is some very beautiful plots as well of that landscape. And we can see in this particular case, it is a very non-linear landscape. But again, you need to use your scientific gut and intuition when you're looking at data like this to say, well, do we really think that these peaks and troughs over in the right-hand graph are real at the very base level? They could just be artifacts in the model. But of course, we can always come back and validate our model and use our model to predict new condition sets that we can run and then use to compare against our model prediction. And in this particular case, that's what we did. So we can see the original actual values against our original model predicted. And then all of the dots in red here were conditions that were predicted by our model that should fit somewhere along this line. So we wanted to predict a whole set of conditions that would sample across our response landscape. And you can see from this one experiment that we got highly detailed models that allowed us to predict what would happen in our system if we were to change one parameter or another. Now, of course, as you start to get to the high end or the extremities of the model where you have less data, then extrapolation of the model isn't as good. It's not as predictive, but that's to be expected with fewer data points. But still, the model performed extremely well. So on that note, a quick summary. I hope from this you can see the DOE really can be applied to anything. So if anyone sat there thinking, well, is this relevant to me? It can be applied to anything that you are doing in R&D. The strategies that you have in your hands is really dependent on the systems that you're running and how much knowledge you have up front as to how many iterations you feel like you need to do, the types of designs you need to do, depending on what your data is looking like. For those different scenarios, so whether you're doing factor screening or you're trying to do an optimization, there are different types of designs that you might use for those different stages in your strategy. But the one thing to always remember is that whilst DOE is a really powerful strategy, a really powerful tool, you still need to be a scientist. Dive into that data, eyeball the data, make inference. You don't always need the models up front straight away. There's a huge amount of learning you can take. And so sort of speaking very quickly to one of the things that Marcus said, best way to get going with DOE is just get going, just jump in and give it a go. It's really not that scary and not that difficult. And I'm sure you'll have fun as well. You get some great data out of it and it's really great to play with. So lastly, I'd just like to end with some acknowledgements. So a big thank you to everyone at Synthace and our partners at University College London and Oxford Biomedica for contributing the data to today's presentation.